along with this. And this blood vessel, from the blood, glucose enters into the astrocytes. There are tight junctions of the astrocytes with the blood vessels, the capillaries. Now, when the glutamates are some excitatory neurotransmitters which are released from the presynaptic membrane. This is a presynaptic neuron, and when the glutamates are there in the presynaptic membrane, it is there nearby in the extracellular space also. And from this fluid, these glutamates are taken up by the astrocytes. Because the glutamates are attached to the postsynaptic membrane for some reason, we will discuss later on, because to create some excitatory postsynaptic potential, or so that to transfer the information or the action potential by the chemical, because the electric current is converted into chemical energy, and that chemical energy, when it is attached to the respective glutamate receptors like the MPA or NMDA receptors, then the threshold of the postsynaptic membrane, which is about minus 70 millivolt, it is a little bit, it changes and it comes out, it comes up. The threshold is lower, and after the glutamates are utilized for initiating the action potential in the postsynaptic membrane, those glutamates are taken up by the astrocytes. These astrocytes now, they will convert these glutamates into glutamine. Glutamine is the primary amino acid. So this glutamine, by glutamine synthetase, enzyme is there in the astrocytes, and it takes some amount of energy, ATP. That ATP comes from the PCA cycle. The glucose which are coming from the blood vessel, that glucose is converted to pyruvate, and as the energy or the ATP comes out, that ATP actually responsible is helpful to convert this glutamate to glutamine, the precursor of glutamate. Also, along with the glutamate, sodium enters. So these sodiums also have to be pumped out. So other energy, other part of the ATP is utilized pumping out of the sodium to make the balance. Now, this glutamine again is released into this extracellular fluid. And this glutamine, which is released into the extracellular fluid, that is taken up by the presynaptic membrane, presynaptic neuron. And this glutamine is taken up, and this glutamine again converted to glutamate by glutaminase, that is in the presynaptic neuron. This glutamate now is ready to be taken up by these clear small vesicles. There are clear small vesicles in the presynaptic membrane which takes up this glutamate, and this glutamate, after taking up, these clear vesicles are translocated. This translocation of these clear vesicles, and finally docking this after the translocation, the docking to the presynaptic membrane. And actually the docking is done with two bonds, two hands. Those are synaptobrevin and syntaxin. These bonds actually attach these clear vesicles with the presynaptic membrane. And then after this, first it is primed with ATP, and then it is docked. And after docking, there are pores for the calcium. So the calcium which is outside, that enters. When the calcium enters, it triggers release of that glutamate into this synapse. Sometimes it happens that there are small pores in the presynaptic membrane. So instead of opening up new pore, that vesicles just open up release the glutamates and then go away. Like it is typically it is called as kiss and run, and they go, and then again it is translocated the empty vessel, and that empty vessel again it is prepared, and there is budding after the endocytosis. This cell wall is again taken up, and then again it is again recycled. So this is the glutamate cycle. So the astrocyte has a great role here. Now the nerve cells are there. There are nerve fibers. We know there are basically three types of nerve fibers we have discussed yesterday. One is thick, it is the large diameter, the diameter of about it is 15 to 16 mu, that is of a lymphocyte or neutrophil. And then this A fiber, this is called A fiber. These A fibers are large diameter fibers. As they're large diameter, so they have less resistance. So they can take the information very fast. They're thickly myelinated, and this myelination is also is done by the Schwann cells. Now, A nerve fibers are again four types: A alpha, A beta, A gamma, and A delta. A alpha, practically, there is a muscle that takes the information from the muscle spindles, and from the muscle spindles, the A alpha nerve fiber takes up the information. And also, there are also Golgi tendon organ, which is from the tendons. When the tendons are stretched, there is configurational changes. Similarly, when the muscles are stretched or contracted, there are some configurational changes in the receptors, like the muscle spindle or the Golgi tendon organ. And we know this mechanical pressure is converted into electrical pressure by the piezoelectric current is generated. So this piezoelectric current which is generated, that initiates the action potential. So this receptor, which is basically transducer, which generates some energy or electric energy, that is responsible for the initial part of the action potential. Similarly, so this is about this Golgi tendon organ and also the muscle spindles. Similarly, there are some receptors which are very sensitive to touch. Touch means less pressure. So if it is in the skin, this is the epidermis, this is the dermis, and this is the hypodermis of the subcutaneous tissue. In the subcutaneous tissue, the nerves and vessels are there, and in between the dermis and near the epidermis, there are some special receptors which are called Meissner's receptor. These Meissner's receptors are also sensitive to pressure. This also generates that piezoelectric current, and from that current initiates action potential. Then there are some receptors like the onions, shells of onion. There is a Pacinian receptor. This Pacinian receptor, as the Meissner's receptor, they carry light touch, fine touch. So it is superficially placed. And the Meissner's receptor and the Pacinian corpus is deeply placed. It is at the hypodermis, practically below the dermis. And so you need more pressure to configure or to distort this receptor. So the action potential or membrane potential is generated with more pressure. So it is with the pressure and it is the fine touch. So all these informations go fast with these A fibers, and this is A alpha and A beta fibers. There are also many other receptors like Merkel disc. Merkel disc is a flattened disc. It also acts on the crude touch and the pressure also, and also stretching of the skin. So that is also taken up by these A beta nerve fibers. Now these go towards the through the peripheral nerves towards the spinal nerve. Now we should need a little bit. We now know the generation of the initial receptor potential, membrane potential. Now how the action potential. So we come to here. You can see that there is a, this is a nerve fiber or cell. Say for example, cell. Inside the cell, there are lots of ions. There are positive charge sodium, potassium, negative charge chloride ions. There are organic anions too. Now, this sodium concentration is inside the nerve cell, inside the cell is about 15 millicovalent per liter. Whereas outside it is 150. It is many, much more. Potassium it is reverse. Inside it is about 140, 150. But outside it is 5.5 or 6 or like that, 5 to 6. The chloride inside is very less, about 9 to 10. And outside it is 120 to 125 millicovalent per liter. So you can see the concentration gradient is different in those ions. So why the ions move? How the ions will move? We know the nerve fibers are normally very easily permeable to smaller ions like this chloride and potassium. The potassium, as the concentration gradient is more inside, and so it has a natural tendency to go out. So potassium efflux, it actually lowers the this membrane potential or lowers this membrane potential further. It makes it further negative. But as the inside of the cell, there are lots of anions, these organic anions or these proteins. So they will not allow. They will not allow. So that's why these cannot go out, because this electrical gradient is negative inside. So it will try to retain the potassium. So potassium cannot normally go out very easily, though it can go. The pores are very free. But what happens to chloride? Chloride is more outside. So chloride has a natural tendency to go inside. So it will create more negativity inside. And as it creates more negativity, ultimately it reaches at equilibrium at about -70. At -70 milliequivalent, it reaches equilibrium. So it cannot move further inside. What about sodium? Sodium has a natural tendency to go inside. But because the concentration gradient and electric gradient, this both are in favor of sodium to go in. But these pores are very resistant. They will not allow sodium to go in. And that's why it is the pores are normally they are closed to sodium. Because if the sodium goes in, this negativity will go. It will become positive, and the membrane potential will go up. The threshold will be lesser, and so there will be stimulation and action potential will be started. You know, from 70, it is normally at 70, it is the resting membrane potential. So if you place an electrode inside the nerve fiber, a microelectrode, so you can calculate the membrane potential. So inside the membrane potential, it is resting membrane potential is -70 millivolt. If you give a stimulus, nothing will happen sometimes. But you give repeated stimulus and like that pressure from the piezoelectric force or anything, current. So after that, after latent period, it starts. So it starts going up. And if the pressure is enough, or if that electric current is generated enough, about this 15 millivolt, then it reaches the firing level. And when it reaches the firing level, the sodium pores are opened. And so the sodium now is open and sodium will go in. So inside will become more positive. So it's rapid ascent. This is the action potential and it goes up rapidly. But as you know, the sodium can go up even up to more, maybe plus 60 millivolt. it reaches at the equilibrium, but as it is very short lasting. This opening of sodium is not long lasting like potassium or chloride. So it again closes very fast. Within maybe 1 millisecond, it stops. And so as it closes, it can reach up to that level. Even if it can go, the doors become closed. At the same time, when it reaches at the zero potential, then potassium channels also open. This potassium pore, the potassium starts going in. And as the potassium, the potassium starts going out. So potassium efflux, this is the natural tendency. The potassium goes out. And if the potassium goes out, the sodium cannot move that high. And so it makes again the negative, the action potential again is lowered by the efflux of the potassium. So potassium comes out, and ultimately it reaches that firing level. So this is called repolarization. Depolarization was due to sodium. Repolarization is due to potassium efflux. But this potassium goes out, and we know the equilibrium of potassium reaches about -90 milliequivalent. So it goes, and this is after depolarization. It is not very stiff, and then it goes further down. And then what happens? The chloride ions, they also want to, they come out. And also what happens? Now the sodium potassium pump place. So the sodium is taken out, and the potassium goes in, and also at the same time the chloride goes out. Finally, as a summation, it reaches again the resting membrane potential. So we know this is how that electric current or the action potential goes in a saltatory fashion in the large myelinated nerve fibers, myelinated nerve fibers. These nerves which are taking, so the senses which are taking fine touch, which are taking the joint sense, which are taking the sense of position, which are taking automatic gait. So these informations goes up to the peripheral nerve to the dorsal or ventral ramoid to reach the trunk, and at the DRG or the dorsal root ganglion is the nerve cell of the first order neuron, and the central process enters into the dorsal horn, and they again go through the dorsal region to the dorsal column, goes up. From the lower fibers, that is from the leg, it goes medially. And as the upper fibers are added, they push the lower fibers medially, and they remain lateral. So there are basically two tracts or two funiculus which goes up. This is called funiculus gracilis and gracilis funiculus, or the funiculus cuneatus, from the upper part and the lower part respectively. We know that this dorsal column takes away the high velocity information. So it is an expressway, and like other expressway or the bypass or the expressway, they do not take much variation in the speed. So only this few impulses or the information or the sensations which goes very fast, they are allowed there, and they travel very fast, go out, go up and up, to reach finally at the upper part of the medulla at the nucleus cuneatus and the nucleus gracilis, they terminate. So remember, the first order neuron starts from the skin and or the deeper tissue or the muscles or the tendons, and they end up at the upper part of the medulla, nucleus cuneatus and nucleus gracilis. From there, second order neuron starts, and crosses the midline. They goes to the opposite side, and then they again ascends, like a flattened column. They have a flattened tract which is called lemniscus. This is called medial lemniscus. This medial lemniscus, to the medial lemniscus, they end up at the ventroposterolateral part of the thalamus, VPL. And from the thalamus, third order neuron, they emerge and they go to the posterior limb of the internal capsule. Internal capsule is basically within this lentiform nucleus and thalamus at the upper part of the midbrain, and from the posteriorly they fans out to reach the posterocentral cortex and like a crown. So this is called corona radiata. So this is about high velocity information going through the dorsal column medial lemniscal system pathway. Now what about the information for the pain? We know for the pain also in the nerve there are two types of receptor or free nerve endings. Free nerve endings, one is associated with A delta thinly myelinated nerve fibers, A nerve fibers, and also a little bit deeper the C fibers. Because both this A delta and C, they carry not only pain but also they carry this temperature. And this temperature and pain and the cold is also carried by this free nerve ending, the A delta fibers. There are also special, there are many receptors which are stimulated, which generates the membrane potential, alters the membrane potential, lowers the threshold. It is like CMR or that is a menthol, cold and menthol sensitive receptor. Also the TRPV1, that is transient receptor potential for the temperature, and these informations they changes the membrane potential and the action potential similarly generated, and that nerve fibers will take the information that is slower. They will go to the same route at the DRG, then they will enter the dorsal root horn, and at the substantia gelatinosa, they can synapse with the second order neuron. Now remember, in the touch and those A nerve fibers, A alpha or A beta nerve fibers, which never ended here, they never crossed the midline. Actually, they cross the midline at the medulla. These fibers will cross here. The A delta fibers will cross with less number of neurons to the lateral spinothalamic tract, and while the C fibers will then again go through many routes, their branches, and finally they also end in the lateral spinothalamic tract. The C fibers, as there are many branches or roads, so you basically cannot localize where from it is coming. But A delta, the sharp pain, pinprick pain, that is precisely localized. You can get the information. Now, these nerve fibers, these postsynaptic second order neurons, they go up along with some fibers of the crude touch. Those crude touch, they also comes to the anterior, this anterior spinothalamic tract. So they also join the lateral spinothalamic tract. Similarly, there are spinotectal pathway from the spinal cord. It goes to the tectum at the midbrain to give the information. So this is to create the spino-visual reflex. So all these three nerves, they go up, and at the level of this medulla, these nerves, they are again comes into another bundle, and that is also another flattened bundle that is called spinal lemniscus. And this spinal lemniscus nerves also, those bundle also goes up. That is the second order neurons, and this spinal lemniscus from the A delta or the first nerve, they actually end in the VPL, because we know most of the sensations end at the VPL, and the crude touch also ends at the VPL. But the C fibers, which carry the slow throbbing pain, they also send some information to this area, the midbrain and this brainstem, that is the reticular formation. This reticular, at this reticular activating system, a RAS, which is basically switch station of the brain, they give information to this RAS, so that even say for example in the night when we're having any visceral pain or cardiac pain, we wake up, and from this reticular activation system, this information, these nerves again go into the intrathalamic nuclei, and from the intrathalamic nuclei again to various parts. Another pathway is there. So in the posterior sensory cortex, we get the location and the intensity of the information, sensory information. It is not pain. It is the sensory information. That is not pain. So unless they're connected to the also this area of the temporal lobe, superior temporal lobe, in between the junction of the parietal temporal lobe, there is the insula. That is the center of the autonomic response. So it is connected to the insula, and so our adrenaline secretion and other things will go. So the heart rate will rise, and sweatings and other stress, the autonomic response will come. It will become unpleasant, and also they're connected to a special area here at below this frontal cortex, and this is called below the parietal lobe. This is cingulate cortex. The cingulate cortex normally it is a area of the destination of the emotion. Normally you are in a happy mood, but when the information goes from the spent fibers, you try to start crying. So the emotional component, so the pain of the definition, why is ISP? They're telling that it has got some emotional experience, it has got some unpleasant sensation and the sensory information. Altogether, the pain is basically the output. Pain may not be in true sense truly sensory information. Based on the sensory information, the output is pain. Now it is about the ascending path for the pain and other things. Again, now we have to talk about the descending path. For the descending path, you know, in the brain there are some special nerve cell bodies near the ventricles. This is the lateral ventricle, this is the third ventricle. The periventricular gray. This periventricular gray, when the nerve goes, the pain fibers goes up, it is connected with this periventricular gray also. And it is stimulated, and periventricular gray liberate endorphins and enkephalins. And along with the periaqueductal gray, they also secrete endorphins and enkephalins. And to stimulate a special area at the brainstem, that is called a special area nucleus raphe magnus. This NRM or the special nucleus, they are stimulated by those nerve fibers coming from the periventricular gray and periaqueductal gray, which release endorphins. Any problem? So this is any problem. So this is now these from the this nucleus raphe magnus, they also send the serotoninergic nerves, which goes down, and they actually send some information or send some process to stimulate the interneurons. This serotonin, which is liberated here, they will actually stimulate the interneurons, which will inhibit both the presynaptic and the postsynaptic membrane. That they will release again enkephalin there, and that enkephalin is attached to the mu and kappa receptors at the presynaptic and the postsynaptic membrane. If it is the presynaptic membrane, it blocks the calcium entry, and if the calcium entry is blocked, so glutamates will not be released. And this is how descending analgesia system acts even whenever there is a little bit of pain. At the same time, the ascending pathway, which is for the slight touch, it goes up, it sends a central, it's a lateral tweak. It is a modified, magnified area. You can see the pain fibers are going and the second order neurons, and those nerve fibers which are taking light touch, they send one tweak, lateral tweak, which stimulate or excite the interneuron to release glycine. And this glycine will actually make the chloride ions efflux, they will influx. So chloride will go in, potassium will come out, and as a result, the information which was excited by the glutamates, that will be inhibited. So these inhibitory interneurons. And contrary, the descending pathway, they actually inhibit both pre and post synaptic memory. And these ascending, this touch information, they actually inhibit the postsynaptic nerve fibers. So this is how pain is reduced. So we can know that there is ascending pathway and there is descending pathway and there is ascending modulating pathway, which is called gate control theory. So this is in a nutshell what there are. Do we have time? Huh?
Less than 1 minute also.
So we have almost finished. So we also know a little bit of the motor areas. This is an interesting. The EPSP and IPSP, excitatory postsynaptic potential and the inhibitory postsynaptic potential. What is that? From the we know when a muscle is stretched. When a muscle is stretched,